I would like to quickly show you my disclosure slide and then introduce to you this 3-year-old boy who developed periorbital edema, total body edema, and ascites, which was due to nephrotic syndrome, which was steroid resistant. And his renal biopsy showed that he had focal segmental glomerulosclerosis, as you can see here, where these fiber strands have replaced some of the functional glomerular capillaries. In nephrology, often we have very complicated names for specific diseases, but often we do not know the etiology or primary cause of the disease. However, in this patient, we are able to know exactly why this child has to go through all the ensuing clinical course, which he had to be entered into the chronic hemodialysis and transplantation program. And this cause of his disease is shown in this slide because he carries a homozygous point mutation in the Potosin gene, which is also known as the NPHS2 gene. And I'm comparing here two partial sequences. The upper one is the normal and the lower one is from this patient, where you can see the 4 different colored nucleotides in the upper row, and then the triplet codons, how each 3 encode one of the amino acids that code for the Potosin protein. And in this patient, a cytidine is replaced by a thymine in this point mutation here, which changes the triplet codon, which then encodes not an alanine, but rather a valine. That changes the structure of Potosin and thereby its function and causes FSGS in this child. And a situation like this, in my mind, is one of the strongest cause-effect relationships that we can see in clinical medicine, probably short of trauma, in that a mutation in one base pair of the 3.3 billion base pairs that we have in our genome is sufficient in and of itself to cause disease, and in this boy, FSGS. However, a situation like this is characteristic for thousands of diseases that we call monogenic diseases, or Mendelian disorders, or single gene disorders. And in nephrology, over the last 10 years, over 250 of these have been discovered and described. And monogenic disease is defined by the fact that in a given patient like this boy, the disease is caused by a mutation in one gene only of the about 20,000 genes that we have in our genome. A disease gene can be recessive or dominant, where in a recessive disease, both copies of virtually all of these 20,000 genes that we have, one from father and one from mother, need to be mutated in order for the child to develop disease. And that's, for instance, the case in ARPKD, where the parents are healthy heterozygous carriers. They have one mutation but one healthy allele and will never develop ARPKD. And in contrast, in a dominant disease gene, if one of the 2 copies is mutated, disease will ensue, and then often one parent is affected, and the disease will have traveled with this heterozygous mutation throughout an entire pedigree. So the term monogenic, however, does not exclude that in different patients, different monogenic genes may cause a similar disease. For instance, FSGS can be caused by Potosin or nephrin mutations, but in each patient, it's sufficient that only one gene is mutated. And right now, in fact, there is 47 recessive and 8 dominant genes that cause steroid-resistant nephrotic syndrome with the histologic feature of FSGS. Now, if we think about what is so special about monogenic diseases, let's look at the flow of genetic information as formulated by Francis Crick more than half a century ago in The Central Dogma of Molecular Biology, where he says that a gene is expressed as transcribed into a messenger RNA, which still has the triplet codons, is then translated at the ribosome into a protein, which is the amino acid sequence. And then the proteins, I think proteo in Greek means I'm the boss, I'm the most important one. The proteins determine much of our body's function, the so-called phenotype, healthy or diseased. For instance, if you look at your skin, you're looking at the keratin protein, plus some lipids. Insulin is a protein. Receptors, transporters in the kidney are proteins. Now, in a monogenic disease, the single gene mutation represents the primary cause of disease or its etiology. So we can say exactly why this patient inescapably developed this disease. And once we know that gene and its mutation, we have a secure entry point into this black box that we're often looking at as nephrologists of the disease mechanisms of, for instance, FSGS, because we know in this patient, FSGS develops because of this mutation. It then can take decades actually to figure out what goes wrong. Even in ADPKD, we now have known the gene for 25 years almost, but we still cannot know exactly how the cellular defect leads to disease. But at least we have a secure starting point and do not need to speculate about disease cause. So the consequences from knowing this specific monogenic disease gene mutation are that we can provide the patient and the family with an unequivocal molecular genetic diagnosis. We have a secure starting point to study the pathogenesis, and many disease mechanisms have been revealed in this way. And we can already draw genotype-phenotype correlations where, for instance, truncating mutations that abrogate the entire function of, say, podocin lead to early onset FSGS versus missense mutations that exchange this one amino acid, like in this boy, may lead to later onset of FSGS. And I'll show you more detail on that in a minute. That's called a genotype-phenotype correlation. And it's already begun that knowing these, we can actually engage in personalized therapy for some of these patients. Just to compare one more time, recessive monogenic disease genes in the first column with dominant ones, and then with a polygenic situation where before disease occurs, the action of defects in multiple genes has to come together. Genetic causality is very strong in recessive diseases because the patients who have both alleles mutated will 100% at a certain age develop disease. In dominant, it's weaker because it ensues later. And there is something called incomplete penetrance, which I'll show you in a minute. The predictive power of identifying the mutation is very strong, therefore, in recessive diseases. And the prognostic power, it's very weak in polygenic diseases. The onset of recessive diseases has a tendency to occur earlier, whereas dominant mutation diseases often occur in adolescence or adult life. These genes, the recessive and dominant ones, have been discovered by whole exome sequencing, copy number variant analysis, whereas polygenic disease genes are often mapped to a specific chromosomal region by genome-wide association studies. And whenever you see the term association, that means there's no clear causality that a specific mutation is known to inescapably cause disease. And these association loci often show a weak relationship between this gene locus and the likelihood or the penetrance of disease. Animal models are very feasible in mouse knockout, zebrafish knockout for recessive diseases, and are difficult to generate for polygenic diseases. Recessive diseases are very rare. Polygenic diseases will probably turn up to be, or polygenic causes to be more frequent, but it's still very hard to show cause-effect relationships there. So more specifically, I'd like to show you age-related penetrance and the genotype-phenotype correlations for recessive monogenic disease genes and dominant ones. Here we have a theoretical cohort of 100 patients, which is the 100%, who manifest with, say, nephrotic syndromes, steatosis nephrotic syndrome, between birth and 100 years old. And usually these onset of disease curves are S-shaped and have to have a characteristic that is the 50% age of onset, for instance, in podocin mutations. If a patient has a homozygous truncating mutation, that means both copies of the gene are truncate the protein so that there is zero function for this protein. If another patient has a mutation in the very same gene, this curve may be bent over to the right, and there may be late, on average, and later onset of disease, and that's actually the case in truncating mutations in podocin versus missense mutations that lead to early versus late onset FSGS. Dominant diseases have a tendency to manifest in adulthood, and they have this phenomenon that's called incomplete penetrance, where when you draw the pedigree, certain individuals will have the mutation but will never develop disease even in old age. And it's an ill-understood mechanism, but it applies to dominant diseases, not recessive ones. But again, there is genotype-phenotype correlation where mild mutations may shift this curve over to the right so that some patients may not live to see the onset of their disease, luckily. Now, I don't want to confuse you. Mendelian disorders are beautifully simple. Recessive means you have to have both copies mutated before disease occurs, and heterozygotes, as a rule, Alport is a big exception, heterozygotes remain healthy. And in dominant diseases, one copy, with a few exceptions of incomplete penetrance, is sufficient to cause disease, unfortunately. There are a few cases or genes, however, where depending on the specific mutation of the same gene, it may behave in a recessive way, where you have to have two mutations, or in a dominant way. But I don't want to confuse you. For instance, some nephrolithiasis genes do these kinds of things. So now Mother Nature can make things worse. And even though patients have the exact same mutation in a gene, a monogenic gene, certain environmental influences can lead to earlier onset. And we hope by understanding the mechanisms that we can develop treatment and prophylaxis so that we can mitigate the onset and shift these curves over to the right. So, quick boards question. The term monogenic disease means A, it is synonymous with single gene disease or Mendelian disorder. B, it describes autosomal recessive, dominant, and X-linked disorders. C, it describes a genetic association. D, it means that a mutation in a single gene is sufficient to cause disease in an individual. And I would personally say A is good, B is true, D makes sense, but C refers to association studies where there is this weak relationship between gene and manifestation of disease. So let's go with E. Now, we asked ourselves 7 years ago, when many of these disease genes were found for FSGS, what percentage of children and young adults with chronic kidney disease actually have FSGS because of mutations in one of these genes? At the time, there were, let me first show you this slide for not only FSGS, but other disease causes of chronic kidney disease before the age of 25 years. I restrict it to this age group because I'm a pediatric nephrologist and that's where we do the studies. So in children and young adults, 50% of the patients develop chronic kidney disease because they have congenital anomalies of the kidneys and urinary tract, in short, CACUT. 15%, like this boy, have steroid-resistant nephrotic syndrome. 14% have chronic glomerulonephritis. This includes MPGN, Wegener's, but also atypical HUS here. 6% have cystic kidney diseases such as ARPKD, ADPKD, nephrolithiasis, medullary cystic kidney disease, and 3% have nephrolithiasis plus minus nephrocalcinosis. So 88%, these 5 colors, fall into one of these 5 groups. And until recently, over 230 and now actually 250 monogenic causes of any of these causes of chronic kidney disease before the age of 25 years have been discovered. There is 35 genes for CACUT, 55 for nephrotic syndrome, 10 for chronic glomerulonephritis if you include atypical HUS, 100 for cystic kidney diseases. That includes ARPKD, ADPKD, 20 genes for Bardet-Biedl syndrome, another 20 genes for pure nephrolithiasis, genes for Alström syndrome, genes for Joubert syndrome, and so on. And there's 30 genes that are monogenic causes of nephrolithiasis and or nephrocalcinosis. So altogether about 250-ish. Now, if you ask what percentage of these patients, in what percentage of these patients can we find the causative mutation in any one of these 230 genes? And we've done studies in virtually all of these disease groups in the last 5 years. Then currently it stands at about 17% for CACUT. If you look in these genes, in 17% you can find causative mutations. In nephrotic syndrome, this is between, maybe in the US, 12%, and in the Mediterranean, 30%. In chronic glomerulonephritis, it's probably around 14%. Cystic kidney disease is 70%. You can find a causative mutation in one of these genes. And in renal stones, it's probably, this may be an over-exaggeration because it was only in a certain cohort, somewhere between 7 and 20%. So we asked what percentage of steroid-resistant nephrotic syndrome is caused by a single gene mutation, and you can actually find it in these patients. We ran a study of 1,780 cases with steroid-resistant nephrotic syndrome worldwide, all with onset before age 25 years. And at the time, there were 26 genes, which we examined with a high-throughput PCR technique. And what we found was in 30% of these individuals, we can find causative mutations. 10% had podosin mutations, 7% nephrin mutations, 5% Wilms tumor 1 mutations, 2% phospholipase C epsilon 1 mutation, 6% then shared into 22 other genes. And it is currently keeping going like this, where very, very rare genes add to the number of causes that we can identify as causing steroid-resistant nephrotic syndrome. So, as I said, this high percentage, however, has to be taken with a grain of salt because it's age-dependent. So if we look in this cohort at age of onset from birth to 17 years, then in infants and newborns, we can actually find a causative mutation in 60%, in toddlers in about 25%, in school children in about 15%, and in young adults in 10 to 12%. The likelihood of detecting a causative mutation is also geographically dependent because it depends on the frequency of consanguineous marriages throughout the world, which I think on average is 10%. And what I'm plotting here on the x-axis is the percentage of consanguinity in these 5 groups from 0 to 100%. And then on that was the y-axis. On the x-axis, the families in whom we detected a mutation in any one of these 4 colored genes, podosin, nephrin, WT1, and other genes. And you can see in areas of the world where consanguinity, where the parents are related, is more frequent, there's a risk for recessive diseases because the same mutation can travel to the father's side and mother's side to the child and lead to the fact that both alleles are mutated. And actually, I think we all have about 10 very, very bad mutations in 10 different recessive disease genes, but we are heterozygous, so nothing bad will happen to us. And we don't know it unless we have sequenced our exome or our genome. But that implies that if there's consanguinity, there's a likelihood that one of these alleles from a common ancestor to mom and dad will come together in the child and cause a recessive disease. On the other hand, if we now look into recessive disease genes, they will often appear sporadic in your clinic. And here's why. My friend Gregory Mendel stated that if parents are heterozygous for a disease gene mutation, and they have 4 children, there's a 50-50 likelihood that one or the other chromosome will travel to the children. Blue is from the dad, green is from the mom. X means mutation, no X means healthy allele. So you can see here 2 of the children will inherit the mutation from the dad, 2 will inherit the mutation from the mom. And statistically, only 1 child will have both alleles mutated and have the disease. 2 children statistically will be healthy heterozygous carrier, and 1 will not even be a carrier. So in a recessive disease gene, before there is familial occurrence, you have to have 8 children because then 2 of them will carry the disease. So in other words, if you go to your clinic with 10 children and young adults under 25 who have steroid-resistant nephrotic syndrome, this is happening in the US, and there's a 12% likelihood that a child could have a monogenic recessive disease mutation. If you take the history in all 10 families, family history is completely negative. That doesn't preclude at all that in 12%, maybe one of these patients, you can find the causative mutation because parents will be healthy in recessive disease genes and every ancestor will be healthy because at worst, they're heterozygous. So Monogenic diseases, if they're recessive, can occur sporadically, and it does not exclude at all that there could be a monogenic cause. But now let's get back to steroid-resistant nephrotic syndrome and its pathogenesis. Here I'm showing from the Netter Atlas the 4 layers or 3 layers of the glomerular filter, the endothelial layer fenestrated here in yellow, the glomerular basement membrane in green, the mesangial cells in blue, and the podocyte in red. And for decades, it was thought that the mesangial cell is defective in steroid-resistant nephrotic syndrome, but it was the identification of monogenic causes of chronic kidney disease, of nephrotic syndrome, such as nephrin and podocin, that showed that's actually the epithelial cell, the podocyte, that relevantly expresses and uses these genes for its function. So the podocyte, as you know, is octopus-like, has these primary processes that shoot off 90 degrees secondary processes that again shoot off 90 degrees tertiary processes, which are also called foot processes. You can't see them here, they're very small. But if you cut through them and hugely amplify it in the next slide, then you see the glomerular basement in green down here. Down below is the capillary, and here's the huge podocyte sitting with its foot process, neighboring another foot process from another podocyte. So they form between them the slit membrane, which is essential for glomerular function in keeping corpuscular elements and protein in the capillary and preventing them from being filtered. The red bubbles that I drew are proteins that, if the genes are mutated in a recessive way, FSGS will ensue. And the blue bubbles are proteins that are encoded by dominant FSGS disease genes. So these are the about 55 or so, and fascinatingly, they have been realized to be part of protein-protein interaction complexes, which I drew boxes around here. And when you look at what these protein-protein interaction complexes are thought to be doing, you can assign function. There are CoQ10 biosynthesis enzymes, there are even nuclear porins, there are RORAC CDC4 regulators, there are the slit membrane components, nephrin and podocin. And let's not forget, each one of these proteins is absolutely necessary for prevent the patient from developing steroid-resistant nephrotic syndrome, because if it's mutated, this disease will develop. And in this way, we have an understanding of the complexity and the components of podocyte function and the pathogenesis of nephrotic syndrome, which in one case may already or has led to treatment consequences. I'm showing you this example here of a 6-year-old girl who had steroid-resistant nephrotic syndrome. She had an episode of a remission, and since she had a mutation in one of these coenzyme Q10 generating enzymes that had low coenzyme Q10 in her tissues, she was giving this, what is pretty much a food supplement and has no side effects, in an experimental way. Then the parents forgot to give it, and she had a relapse, and then she responded. So for patients who have mutations in ADCK4, COQ6, and so on, this is a treatment where there are 4 papers now that show that if started early, it can at least mitigate the development of FSGS. Now, let me move on to another group of chronic kidney disease. These are the cystic kidney diseases. You know that they've been also addressed as the so-called ciliopathies, because the proteins and genes that are mutated here in these monogenic cystic kidney diseases are part of what is called primary cilia. These are little antenna-like structures that can be virtually built by every cell in the body. Photoreceptors are actually specialized primary cilia. You've seen in the textbooks that principal cells in the collecting duct have this beautiful cilium sticking out, which may have a mechanosensor function. They're very complicated antenna-like structures which have to do with reception from outside signaling and maintenance of cellular differentiation. And similarly to the monogenic disease genes and proteins in steroid-resistant nephrotic syndrome, many of the ciliopathy proteins interact in protein-protein interaction complexes, and define some of these functions. The diseases are polycystic kidney disease, recessive or dominant, nephronophthisis with extrarenal manifestations of retinitis pigmentosa, and in Bardet-Biedl syndrome, as you know, also anosmia, liver fibrosis, obesity, diabetes, and it can also be associated with situs inversus because cilia play a role in that, and with neurologic defects such as cerebellar vermis hypoplasia. Excuse me. We studied a cohort of 79 children and young adults who were included in the study by the sheer criteria of either having increased echogenicity in both kidneys and or cysts. But even increased echogenicity was a sufficient criterion. We sequenced all the, at the time, 30 ciliopathy genes and found causative mutations in almost 2/3 of them. So this is the entire cohort. And the blue segment is the 63% in whom we found causative mutations in one of these genes. And then if we take the blue cohort and make it 100%, then yes, the new blue segment were real ciliopathy genes, nephronophthisis genes, ARPKD, and so on. But the others were so-called phenocopies. And there were a few recessive Alport cases hidden in here, shown here in green. There were some Cocoot cases hidden in here. But in general, you can now say anyone who has a renal ultrasound under the age of 25 that says increased echogenicity, if you jump from this very unspecific comment by our radiologist saying compatible with medical renal disease, to sequencing the whole exome, you have a 63% likelihood of finding the primary cause of disease in one of these genes in these rare diseases. And I want to give you a few examples because sequence all the genes now gives us an opportunity to solve some of these very difficult differential diagnoses. Here we have 5 cases with increased echogenicity plus minus cysts. Here is just a histologic image. You can see there's clear echogenicity, there are cysts, and when we sequenced the exome and we looked in the 30 kinase genes at the time, now it's 100, we found causative mutations in the nephronophthisis type 4 gene, NPHP4. Here actually there was also echogenicity, which I'm not showing you. This was an SLC41A mutation, which is a hypomagnesemia gene. Here you would have said as a nephrologist, looks like pepper and salt, this could be ARPKD, and you would have been right because there was a homozygous PKHD1 mutation. This actually, you see the kidney here, echogenic, has a few cysts. This was AGXT, it was hyperoxaluria type 1, which we very frequently see as underdiagnosed or recognized too late, and which can now be diagnosed early if any patient with increased echogenicity before age 25 receives or is offered a panel or whole exome sequencing, which is very important because early treatment is important in this disease. And these were actually siblings, 2 brothers who had these vastly echogenic kidneys, some cysts. You may have said, oh, there's an increased echo in the pyramids, and you would have been right. This was distal RTA. They had an SLC4A1 mutation in the anion 1 exchanger, and thereby distal renal tubular acidosis. Excuse me. Now, finally, let me move to nephrolithiasis. We showed in 3 papers now that in anywhere between 18 and 7% of patients who have at least 1 stone before the age 25, if we examine 30 monogenic causes for renal stones, we can find a monogenic cause which often has specific treatment consequences. Now, finally, let me move to a brief case. A 7-year-old boy with cystic kidney disease and hyperuricemia, and he also had an atrial septal defect. He had still normal renal function and normal blood pressure, and urinalysis was unremarkable. He had no systemic abnormalities other than the ones described, and at least none that were related to his chronic kidney disease. The additional findings were the hyperuricemia, a slightly decreased magnesium. He was diagnosed with autism spectrum disorder, and he had a positive family in his father, paternal uncles, and grandmother who had a kidney condition. His renal ultrasound is shown here. You see the echo pattern of the kidney is hardly to be differentiated from the liver, so it's a little increased because it should be darker. On the other side, he has again increased echogenicity and potentially a few cysts. This is the pedigree. This is the boy, 7 years old. And there was kidney disease in his father, the father's two brothers, unfortunately, and the paternal grandmother. And everybody had high uric acid. And if you look at the pedigree, this is not compatible with a recessive disease, but with a dominant inheritance pattern where one copy, if mutated, segregates to multiple generations, even if the other copy of a certain gene is normal. It is not compatible with X-linked disease because you have male-to-male transmission, and you know if a father wants to have a son, he needs to give him his Y chromosome and not the X chromosome. So in other words, whenever there is male-to-male transmission of a disease, the disease cannot be X-linked. So there's a suspicion that this would be a ciliopathy or renal cystic disease gene that is dominant, and great candidates are uromodulin, which encodes the Tamm-Horsfall protein, and HNF1 beta, hepatic nuclear factor 1 beta. We looked into uromodulin, which causes medullary cystic kidney disease type 2. The UMOD gene encodes Tamm-Horsfall protein, and UMOD mutations can lead to familial juvenile hyperuricemic nephropathy, or FJHN, but also to glomerulocystic kidney disease. And although medullary cystic kidney disease, or it's called ATKD, I think right now, represents a subtype of congenital anomalies of the kidneys and urinary tract, this is a very, very rare outcome. And we did not find any mutations in UMOD. Neither did we find point mutations in HNF1 beta. However, we found in the exome that there was a run of homozygosity in the region of the HNF1 beta gene where all the alleles were the same, but we know that there's a high frequency of heterozygous deletions. And in fact, this turned out to be a long heterozygous deletion where one copy of the HNF1 beta gene was deleted. I don't want to go, for the sake of time, into this image, but the diagnosis was a heterozygous deletion of HNF1 beta. This is a multi-system disorder. It's a chameleon. It's often difficult to diagnose. It has many extrarenal manifestations, some of which are listed here. One of the most important ones in way of a personalized diagnosis or personalized therapy is to realize that many of these patients develop MODY diabetes type 5, so one has to be on the guard for that. Excuse me. They can have pancreatic hypoplasia. They can have genital malformations, which are listed down below here. They can have elevated liver function tests. They very often have hyperuricemia, like every affected in this pedigree. And they have hypomagnesemia, and of course can develop renal failure. And there is more than 150 different mutations known now, but 50% of the genetic abnormalities are actually heterozygous deletions, as in this patient, and that they are hard to diagnose or cannot be diagnosed just by Sanger sequencing, so one will have to run copy number variation analysis together with the panel that you're running. Often also there is de novo occurrence where the heterozygous deletion will have occurred in the patient only, and his family, his or her parents and everybody in the ancestry will not have the deletion. That's something important to keep in mind. Excuse me, again, as an example that lack of familial occurrence doesn't exclude a monogenic cause of disease. Clinical implications for diagnosing HNF1 beta spectrum disorders can include it provides a definite diagnosis and allows genetic counseling for these families, which can be very important for planning of living-related donor transplantation. It allows avoidance of unnecessary diagnostic procedures because it leads right to the cause of disease. And it allows early detection of extrarenal manifestations such as the hyperuricemia, the MODY diabetes, and it's important to take into consideration if it's actually dominant and inherited when living-related donor transplantation is being planned. So in summary, one can now perform an indication-driven mutation analysis either by ordering renal panels or running the whole exome, which is almost similar in cost by now. And the likelihood of finding a patient's monogenic causative mutation in any patient who develops chronic kidney disease before age 25 is shown here. It's between 12 and 30% for steroid-resistant nephrotic syndrome. For echogenic kidneys, just as a non-invasive renal ultrasound finding, chronic echogenic kidneys, there's a 70% likelihood now of finding a monogenic cause in one of these 100 genes. In congenital anomalies, there's about 17% likelihood of finding a monogenic cause. In renal stones, this is probably a little too high, 7 to 18%. So overall, there's an almost 20% likelihood in this age group to find a monogenic cause for chronic kidney disease, which can have many, many consequences for the management of these patients. And luckily, increasingly, insurers will see that and will pay for that. And if they don't, I would recommend to provide them with publications from our lab, from Ali Garavi's lab in adult nephrology and others around the world who now show that there is a very high likelihood of the possibility of finding the causative mutation in chronic kidney disease in this age group. So in state of the art, every patient with a kidney disease that could be caused by a single gene mutation should have a chance to have this mutation identified. Why? Because it can be done now. 10 years ago it was, or even 5 years ago, it was either not feasible or too expensive. It provides the patient with an unequivocal diagnosis if the mutation is found. It may reveal, rarely though, a potential treatment, but in the case of CoQ10 related mutations, this is already the case. And by understanding more of the etiology, this will be the case for many more patients in the near future. Also for clinical studies, I think it's important now to know the etiology. If that can be up to 1/5 of your cohort, it's very important to know that so that you're not comparing apples with pears in your study. It provides the missing pieces for the pathogenic pathways that are still puzzles to us, and it enables the generation of specific animal models which can now help, for instance, with knock-in models to design personalized therapies. And in these models, it enables us to screen for new therapeutic molecules which we are in dire need of. Thank you very much.